Thank you, Jeff, Steve, John, and Parag, for your leadership of MITili. If this hypothesis is true, that by immersion in novelty, you can upregulate plasticity, then perhaps even in the classroom setting, you could potentially enhance learning outcomes by having the students experience phases of novelty. So that's the title of our work, Enhancing Learning via Novelty Insertion. And this is the wonderful team that I had the good fortune of working with. Annie, Matt, Anna, that Kyle had mentioned in his talk, Lara, Sid, who just celebrated his 95th birthday, Ricardo, and yours truly. So here's our experimental design. We decided to take a MOOC, and quite appropriately, we chose the MOOC taught by John, Intro to Psychology, 900. We broke it up into pieces, and we then inserted novel videos, so very short clips of novel videos or not so novel videos, familiar videos. So a student or a participant in our study would experience this whole lecture by John on material that presumably is new to them. But this video would be interspersed with either novel video clips or familiar video clips. The novel clips were about interesting animals in the wild, true facts videos in case you want to check them out on YouTube. And the familiar videos were about a woman describing how to go about cleaning your kitchen or cleaning your room, which turned out to be a somewhat of an unfortunate choice, as I will mention. So this is group one. So group one is first experiencing the novel insertions and then the familiar insertions. And just to make sure that there wasn't an ordering effect, we also had another group that first experienced the familiar insertions and then the novel insertions. And what we did was to then follow up this experience with an assessment period where the students would be assessed on material that had either followed the novel insertions or the material that had followed the familiar insertions. We additionally recorded ECG and galvanic skin response while the students were going through this regimen. We had 47 subjects, 10 of them participated as pilot participants, 37 as participants in the actual study. And the reason I've broken this up into 25 plus 12 is because 12 of them, unfortunately, fell asleep during the experiment, so we did not include their data. So the pilot participants, we had allowed to have coffee before they came into our lab. And then we decided, no, let's skip coffee. And that had these adverse outcomes where a third of our participants fell asleep during the study. So these are the kinds of raw data we collected, cardiac, EEG, EOG, and GSR. And the results, without going into the details, contrary to our expectations, the novel videos did not elicit significantly different physiological responses from the cleaning the kitchen videos. So galvanic skin response and ECG metrics were pretty much indistinguishable between the two. And it was only post hoc when we were analyzing the data that we realized that for MIT students, perhaps the videos about cleaning the kitchen are just as novel as videos about echidna or the star-nosed mole. So slightly unfortunate, which perhaps masked the effects of novelty versus familiarity. But to our pleasant surprise, even with this reduced distinction between novel and familiar insertions, we still saw that subjects performed marginally better on material that followed novel insertions versus material that followed familiar insertions. And furthermore, This advantage of novelty was especially apparent for material that was presented within 8 minutes of a novel insertion. So this brings up some very interesting ideas about how long the effect of novelty lasts. 2 minutes of a novelty insertion, how long the effects of the increased plasticity might last. So just to conclude, this project, it allowed us to make some advances on a very interesting question, both fundamentally and also from an applied perspective. And even though the results are still in the works, we need to increase the number of participants, increase the distinction between familiar and novel videos. I think the results already hold promise and deserve further investigation. And thank you again to MITili for supporting this work.